membranes. We're going to draw a nice picture and look at some of the structures that make up cell membranes. You'll recall that we've talked about how membranes form in a primitive way from fat molecules in the foam of the primordial sea, and how they can form around bubbles. And we saw how individual molecules can actually become a membrane. So we're going to have to learn about a few of these friends, these molecules. So the first molecule, we're going to draw it as a symbol like this with wiggly tails. Okay? But you have to remember what that stands for. What it stands for is a molecule that looks like this. So we'll just draw him generally like this. O, O, and an H like that. There's the first part. You can draw that. And then attached to the O, there's two long tails of carbons, carbon atoms, like this. And they're different lengths. Some of them are wiggly, some of them are kinked and bent, some of them have double bonds, some of them have single bonds. There's lots of options. And of course, this first carbon has another oxygen stuck on top like this. And then all the rest have hydrogens all around. Okay, I'm not going to draw all the H's, but every one of those little sticks that's sort of empty has an H on it, a hydrogen. So we'll just shortcut like that. So you can see that this part is the circle, and these parts are the wiggly tails. So that's what we mean when we draw this picture. Don't just draw this and say, yeah, it's some kind of weird head with tails. Make sure you remember what it stands for, because it's very important. Okay? The other thing we're going to draw, remember what that name is. That's called a well, it's actually called, I'm not going to get too technical here. It's called a lipid. Okay? Now, in grade 11, you're going to study membranes in more detail, and they'll fill in a lot of the gaps. It's called a lipid. It's actually, in most membranes, called a phospholipid, which means it has phosphate stuck to it. Right around here, there's a PO4 sort of stuck onto this. But we're not going to worry about that. We just want to know the general idea. We know that it's a lipid, right, or a fat molecule. It's a kind of, what we might call a fat. Technically, a fat is a sort of a complicated lipid, but that's good enough for what we want. So the lipid molecules, okay, they're lipids and they're fats. That's what we want to remember. And they look like that. The other kind of molecule, right? Maybe I should wait for a minute to make sure you're caught up. Another shortcut picture we're going to use is for proteins. And the picture we're going to use for proteins is basically just a windy string ball like this. And the reason we're going to use that is because what proteins really are, and I'm not going to draw it as complicated as the other, they're C's and O's and H's, along with some N's and some S's. These are all the atoms that we find mostly in proteins. And what they are is they're little chains of these little small groups of atoms called amino acids. Remember that? The amino acids are small. They're smaller molecules. They're like glucose size, maybe a little bigger. And they have some loops of carbon, and they have some N's and some S's sticking out. And they all stick together to make a very long line or chain, like a string. But then what happens is the string gets all twisty and curly, and it turns into a blobby, three-dimensional, string-shaped object. So that's what we're going to call our protein. The thing you should remember about proteins is because they're a big ball of string, they can be pushed and moved and twisted into different shapes. That's one of the most important things about proteins. They can make a million different shapes. And those different shapes mean they can do lots of different things. So if you had to remember anything, remember that. Okay? And then we're going to draw some little stick figures. we're going to draw things like this with like little tree branches that stick out like that in various ways. And these are going to be called glycoproteins. And they're going to stand for two things, what we call lipoproteins. Now, again, we don't have to get into the detail of what this is. Here's what they are. The word glyco comes from glucose, glycogen, glucose. It reminds us of what kind of molecule? It reminds us of the carbohydrates, right? Whenever you see glyco, you should think carbohydrate. You might want to jot that down. Glyco means carbohydrate. So what this is it's a carb molecule, a carbohydrate joined to a protein. And what does lipo make you think of? It should make you think of lipid or fat. And so what we have down here is a fat molecule joined to a protein. Okay? And remember, carbohydrates come in all shapes and sizes. They're made of C's, H's, and O's stuck together in different patterns. But rather than draw all the little C's and O's and H's, we're just going to draw these symbols that kind of are representative of the shape that they make. Okay? And so those are the three important sort of symbols we're going to be using to draw our picture. All right. Now let's draw the picture. We're going to try and draw a three-dimensional picture on 2D board, which presents some challenges, but here's what we're going to do. I'm going to start with a nice green color. We'll try to color code things so they're easier to see. And I'm going to draw, so pretend this board now is your half a page. You need about a half a page to do this, so get a half a page. Start, just about the middle of the page, or sort of down a little bit lower. Don't start up at the top. Start down here and draw circles like this. Circles. Every once in a while, leave a space. Maybe another space. And we'll leave one more space. It doesn't have to be 3 each time, maybe 4, and then we'll put some more like that. Those are the heads of the lipid molecules that are making the membrane. All right? Now, I'm going to just shift to a finer line, and I'm going to draw the little tails. Remember, they all have 2 little wiggly tails. And you can draw the tails different lengths, and you can draw them with little bends and twists, because if they have double bonds, remember what double bonds do, when you're linking carbon together, they make it bend or twist. So just randomly draw them different lengths, and I feel like it's like a Bob Ross video, Little doers, whatever in your world you'd like to see, draw in some tails. Now remember what those are. They are those special lipid molecules. Now we have to do the same thing underneath, almost the same thing, with the bottom layer. So start a little bit lower, like this, put a couple like this, and I'm going to leave a space right there. I'm going to leave a space lined up with the other space. That's important. Line up this space with that space. Put in some more circles. Maybe we'll keep going here, and we'll put another one sort of right there, and then we'll line up that space there. So some of the spaces are lined up and some are not. You see that? And then I'm going to put their tails on. And their tails, of course, go up because they're making this lipid bilayer. And remember why they do this? Because in the water, the tails don't like the water. The tails are hydrophobic. And so they would rather sort of bunch together in the middle and get out of the water. While the heads, the circle parts, are hydrophilic. They love the water. And the reason for that is they're polar, right? Remember? They're polar, and polar things are easily sort of dissolved in water. Whereas non-polar things are not, because of the magic of the water molecule and its polar bonds and how it has plus and minus sides to it, pulling and pushing on things. So again, that simple chemistry concept is the reason why this bilayer forms like this. Okay, now I'm going to shift to a different color. I'll pick red. And I'm going to draw some proteins, maybe with a finer line. So the first protein I'm going to draw is this one right here. Squiggle around your line, all like, remember, he's a big chain of amino acids. And he's all sitting in there like a big coil of string. Very complicated molecule, bulging out at the top, maybe, who knows, whatever. There. That's our protein molecule. And I'm going to draw another one of them over here. You'll notice he goes right through the whole membrane. That's kind of what we wanted to show. Same with this guy on this side. He goes right through the whole membrane. Wiggly, windy string of amino acids. Sometimes these proteins have more than one string of amino acids even, but we won't worry about that. So there's the wiggly, windy protein. Now these other spots, I'm going to use a different color. These are still proteins. And we can tell because we're going to draw them as squiggly lines. But I'm drawing them a different color because they don't go all the way through. This one is just sort of blobbed here on the top half. See, like that. And one over here is blobbed on the bottom half, like that. And then over here, we got a space for another one. These proteins are sort of like only half embedded in the membrane. Something like that. And we're going to label all these parts after, when we're finished. So there's the proteins. Okay, now to make it 3D, we're going to draw the top of the membrane. We're going to go back to our green balls here, and we're going to draw this sort of off like on an angle, like this, up here, and then we'll go across like this, just like when you draw a cube, right? And we'll draw another angle like this, try to make the angle match the other side, and they'll meet up over here like this. So this is like looking down at the top. Now, if you were looking at the top, you wouldn't see any tails, would you? You'd just see heads, a sea of little heads. So now we just have to go along here and fill in the sea of heads. But every once in a while, I'm going to leave a space like that. Random. It doesn't matter if your spaces are exactly the same as mine. I'll leave another space over there. Okay. On we go. This is just kind of the fun, boring part where it's like meditating, drawing the circular heads, because we can't see the tails because the tails are tucked downwards into the membrane. Maybe I'll leave one more little space there. Fill it all in with circles. Something like this. Okay, good. We're almost there. There, something like that. Well, this part over here looks like it needs a few circles too. Now we left a few gaps there. So we'll do, some of them can be red and some of them can be blue. It doesn't really matter which one. So I'll put a little wiggly protein up here, right? And I'll put another wiggly protein maybe over here. And if it's red, all that means is it goes right through to the other side of the membrane on the bottom, but you can't see that part, right? Because it's hidden. And if it's blue, it means that it's just sort of only sitting in half like that. So we'll put a couple of those blobbed on there. So we can see what they look like from the top, but the bottom part of the picture shows us the cross section, right? It shows us what's actually going on in a crosswise way. Okay, now we're going to add, I'm going to take maybe yellow, because it'll stand out, and we're going to add little tiny blobs. These little blobs are going to be sort of mixed in with the membrane down here like this. They kind of are in between the tails. Okay? And these blobs are in some membranes, not all membranes. These blobs are a special kind of molecule called cholesterol, which what it does is it's a kind of like a lipidy fat molecule. And what it does is it makes the membrane all kind of gooey and liquidy. It helps the membrane sort of stay very gooey. It's liquid. The membrane itself, it looks like a solid thing, but it's not. It's actually more liquidy. In other words, all these molecules we're drawing, they can easily be pushed apart and pushed together, and they can even move. These big blobby proteins can sort of wiggle, just like you would push your way through a crowd. Pardon me, excuse me, pardon me, pardon me. These blobby proteins can actually move along, and they can push between the green fat molecules and change their position. So the membrane is not a solid structure. It's more of a liquidy. I'm just going to fill in my hole here. I don't like the way that looks. It's more of a liquidy sort of in-between type thing. Okay? So you can imagine this moving and shifting and not being a permanently solid. It's not like a piece of saran wrap where it's a solid plastic sheet. It's very fluid. Okay, one more thing to draw, and we'll use our black. And these are going to be the little guys that stick out. They're like this. They stick out of the top, like that. And maybe some of them are attached to the green balls, like I'm doing here, right? So they are what we call lipoproteins. And then some of them are attached to the protein molecules, like this. And this would be I'm sorry, I did that backwards. Now I just realized. Better glycoproteins. I'm sorry, I have to make a correction. I hate that. This is not a lipoprotein. It's not a fat and a protein. It's a fat and a carb. So let me change that. I'm sorry I made that mistake. When you're doing it live, carb plus protein, carb plus lipid. And what I should have wrote was a glycolipid. Glycolipid. That's the combination we want. My mistake. So these are little carbohydrate chains. And sometimes they're attached to a protein, so we call them a glycoprotein. And sometimes they're attached to the fat, they're called a glycolipid. So that's when I draw them here. Some of them are coming out of the proteins like this. And some of them are coming out of the fat molecules like this. And you can draw a couple of them sort of sticking off. There's actually lots of them. They're all over the place, but the picture gets a little crazy if we draw too many. And they're like little trees that are sticking out. So you have to kind of think 3D. And what's important is they're not only on the top, but they're also on the underside or the bottom of the membrane. So we want to draw a couple of them sort of sticking out of the bottom as well. Maybe one on the protein here, and maybe one or two on the, just enough to give us the idea. Okay? They're underneath and on top of the membrane. Okay, so this is a pretty reasonable picture. Just to make the 3D effect look better, we're going to fix, or we're going to finish the other side of the membrane here. So what I have to do is draw some little tails. These guys are kind of on the outside of my picture, so I'll just draw little tails straight down like that. And then I'll draw more little tails going up like that. And then I can go back and draw some little heads sort of going attached to those tails going up this way. There, something like that, just to finish off that side. So there's a little slice of membrane, a little chunk of membrane so far. All we have to do now is talk about what each of the things do. So I'm going to pause for a minute, let you catch up with the picture, and then we'll label it, and we'll talk about what they do. Let's label, and I'll label with a purple color so that it stands out. All my labels I'll do in purple, and then we can, hopefully it won't get too cluttered. Okay, so let's label the green first. Well, actually, maybe here's an idea. Why don't I label in the color that they are? So I'll use a green for this guy, and we'll have to write over here. Maybe we'll squeeze it in over here on this side. Okay, put a little one of these, a little bracket, which is showing a whole doubled up layer of green molecules, and that's called the phospholipid. Remember, I told you phospho just means it has a little bit of phosphate attached to it. Phospholipid bilayer. it's a double layer. You see how the membrane has a top and a bottom, and they're stuck together. And remember, the reason they stick like this is because those tails hate the water. So this form allows the tails to get inside the membrane and not get in the water. Phospholipid bilayer. And that, of course, forms the main sort of matrix of the membrane. Okay. The next thing are the proteins. So I'll use my red color to label a red one. I'll look for a spot where I can squeeze it in. Maybe up here at the top. I'm going to go, or maybe at the bottom. So I'm going to point to this guy. He's called an integral, or some people say an integral. You pronounce it different ways. If you're a math person, you'll say integral. And if you're not, you might say integral. The word integral, though, means within or inside the membrane. And these ones are the ones that go right through. Integral proteins go from top to bottom all the way through. Okay? And these function as gateways. Remember, I told you how these proteins can open up. So maybe what we'll do, because we're running short on time, is we'll label this, we'll call that the end of this video, and then we'll make a second video to explain all the pieces and what they do, okay? So this will be the integral protein. The blue ones are also proteins, but they only stick on one side. So they're called peripheral, right? Peripheral means on the outside, like your peripheral vision is sort of on the outside of your field of vision. Peripheral proteins. Because they don't go all the way through, they're not very good gates or doorways, but they can be all kinds of important identifiers. They can be labels, like license plates that help tell the cell, whose cell this is and what it does. And they can be sites for other molecules to attach to make the membrane do things. So they're attachment sites mostly for other molecules that can influence how the membrane will act. Then we have two more things to add. The black molecules, right? This one I've drawn here would be, because it's attached to the fat, it would be a glycolipid. And these also help identify and give signals as to what kind of cell this or membrane this might be. And then over here, this one attached to the blue, I'll point to him and I'll call him a glycoprotein because he's attached to the protein. All right, and then the last thing are the yellow blobs, which I'll write in yellow. And if we pick a yellow blob and a spot where I can write a big word, maybe right here, we'll point the arrow in. And those are blobs of cholesterol. Now, you may have heard that cholesterol is bad for you. Whoops, I better spell it right. Cholesterol. Cholesterol. Hang on, here we go. Cholesterol. Cholesterol is bad for you if you have too much of it, but it's very important to have. If you're an animal, mostly animal cells have cholesterol inside them like this. But again, that's to help the membrane stay more fluidy and less rigid. But we can talk about all those kinds of things shortly. In the next video, we'll discuss the role of each of these things and what they do. Okay? So then you can call that a day. There's your picture. That's a cell membrane, a typical one.